we really have the pleasure of having Professor Akshay Anand from PGI Chandigarh, India. Is one of the two premier institutions in India, which is federally funded and really hard to get in. And so a ton of people do research as well as clinical care there. So it is my pleasure to invite Akshay from India. He is the biggest spirits with his hand fracture that he accidentally had just before traveling. He still decided to come and attend a couple of conferences and give his lectures. So it's really, spirits to follow. So he's keeping his word right there. Akshay has done a tremendous amount of work in the last 20 years, especially in the last 4 or 5 years. So much so, I think he's been nominated for the highest awards in the one of the highest awards in the country, the Padma Awards. So that is as high as you could reach. And Akshay has been a neuroscientist at heart, a lot of basic science research, and of late doing a lot of clinical research. He's a professor of neurology in the Neurosciences Center at the PGI Chandigarh. And he's also editor-in-chief for Annals of Neurology and also for Integrative Medicine Reports, which is his latest passion towards that. And he's organized, I think during the pandemic, he's done one of the incredible services for the entire world. He organized a series of lectures every week via Zoom before before even the vaccines came about. And that's called the Yoga Scholar Program, is that right? And he brought in people from all around the world presenting their research and combining that science with the yoga and meditation and really giving that firm backing of science to. And after that, all the talks are now archived and really sought after by many people across the world. So I would say that that is one of his biggest services to humanity at large, especially during the COVID pandemic. Now that spurred interest from the government, and they have funded one of his largest yoga sciences center at PGI Chandigarh, which is trying to integrate medicine with the traditional complementary approaches. So he has taken it up, and he's taken it up at a very high level, going to the government and making sure that implementation happens and being an advisor for the special section to the government. So really making changes at the policy level. On top of all this, he has published just about 250 publications and also 4 patents, and published a couple of books, and he's also serving his editorial role. Akshay, it's my immense pleasure to invite you, and I welcome you to come and talk. Right before that, do you have to say something? I just asked Tulsi to speak a little bit about the center, and then get Akshay.
Thank you, Dr. Bala, and welcome everyone. It's so nice to have you all here in person with us today. So I know many of you are joining us online as well. So just very quickly, want to share a bit about our center. As many of you are aware, our center is a multidisciplinary research center based at the Department of Anesthesia, Critical Care and Pain Medicine here at Beth Israel Deaconess Medical Center. We have many of our team members here today, and we have team members joining us from all over the world at our center here in Brookline, Massachusetts. And our director who just spoke, Dr. Bala Subramaniam, is the Ellison C. G. Pierce Endowed Chair of Anesthesia and also Professor of Anesthesiology at Harvard Medical School. Our center is really based in and focused on enhancing human consciousness, cognition, and compassion. And we do this through at the nexus of research, education, and outreach, and really bringing these together in all that we do. So without further ado, we're very excited to have with us in person today, all the way from India, Dr. Akshay Anand, sharing on the topic of mechanisms of memory.
Thank you, Tulsi, for the introduction. Such a privilege as well as excitement that I stand here in front of you. COVID really brought us together. Bala, I met over the COVID pandemic through Zoom. Yeah, as you rightly said, we were all putting all our efforts to put this. The institute, which is northwest of New Delhi, about 200 kilometers of Himalayas. And it's one of the preeminent premier medical institutes, seeing almost 150,000 patients. The institute is the front gate of PGI Chandigarh, and that's the Trauma Center. In addition to a very busy, I bring in a basic perspective of how I look at healthcare, how I look at things. I had some training in the US in the university. Focus was not only cell survival mechanisms, but more importantly, to bring in the concepts of quality assurance, which is kind of unknown in the research world, almost globally. It's a scheme of things that makes your data auditable, archivable, backtraceable, robust. And I think this is also the reason why we are not seeing many of the bench studies at wet labs translate into clinical products, clinical services, diagnostics, therapeutics, etc. So like any other lab, we set up a brief introduction that I will give in this talk would be to spend a couple of tablets like what we have been on. And then where do we stand with respect to memory? What are the memory mechanisms that we know happen? And then what our contributions have been to the world of learning and memory. You all interact, you sit here and speak. As I speak, you listen and you learn a bit and you forget a little. What's going on, what are the chemical and the electrical underpinnings that underlie learning and memory. Throughout the talk, I would try to send some cues for you to think of how we can use these chemical cues or the electrical cues known to govern learning and memory into non-pharmacological or non-drug approach such as. So I will not go much into, although I will show some very complex slides which are known to molecular biologists as a schematic or a molecular mechanism. Try to see the underlying current of prevention studies or meditation research studies, any of the complementary and alternative medicine. So this lab that I went back to India is finds a biomarker discovery program with the help of a clinical colleague, PSF to our lab in the and we model them into animal models which become the tools for giving therapeutics. A lot of what happens in the biomarker discovery aspect of the lab becomes tomorrow's diagnostic tests or prognostic tests, ethic tests, if I may call them. And we of course use lots of therapeutics such as those that come from stem cells, herbs, and more recently from yoga intervention studies. Yoga practitioners and yoga trainers' blood products and how they actually, what changes they bring biochemically and at molecular level, the animals that we have created. This has been the focus of our biomarker discovery program, and I'm sorry, I know you all can listen, you can all hear me, but since we have some online audience, this will help if I speak a little louder. I am a little soft-spoken. So we've been sampling these samples from patients such as ALS, and we've described how the BEGF, some of the angiogenic and neurogenic markers help, whose deficiency helps predict early ALS, which is a fatal disease. And also for Parkinson's disease, we have shown PARK2 gene as mainly responsible for Parkinson's, especially in the place, in the region of the country I come from. I come from northwest of India, a state called Punjab, and most of them are Punjabis or Sikhs, dominated by Sikhs who do not smoke. So this is something that we reported. Both ALS and Parkinson's disease have a dementia component that brings us to the memory and the learning aspect that I just I'm going to delve on. We've also worked on biomarkers that can predict macular degeneration, a disease of the aging. It's an irreversible it's a cause of irreversible blindness caused due to problems in the choriocapillaries that leak into the Brooks membrane and cause wet form of AMD, which is very bad. And there is, of course, the dry form, which is not so bad, but a lot of about 10% of the dry AMD convert into wet AMD. So these have been some of our contributions as far as the biomarker discovery is concerned. And we have then modeled animals. And this is also important because some of you may think of these models as a tool to collaborate and to use them as not only models for drug screening, herbal screening, for neurotherapeutics, but importantly for our meditation yoga studies also. So we use what is known as the Morris-Water maze. It's a neurobehavioral assay which assesses memory loss and also tries to screen drugs that can rescue such artificially induced amnesia, either caused due to drugs, amyloid beta, or any injury, traumatic brain injury. And we use such stereotactic instruments that are available worldwide, using XYZ axis and the Bregma atlas to deliver at a regulated way and in a time-bound fashion amount of drug that we want to deliver to hippocampus or whichever part of the brain that we want to deliver it. We also have a NMDA-induced retinal degeneration model because we have a lot of focus in retina and retinal degeneration, which is another model of CNS or any of the AMD as a model for AMD or for other retinal degenerative diseases. So we are able to basically dissect out the various retinal layers that are disrupted by either chemical injury such as this or surgical intervention by blocking blood, by a stroke model or a variant of that is called PPA, peridopalentine artery occlusion model, and by which we can occlude the blood flowing into the thalamic artery and it can cause retinal degeneration as well. And it's another excellent tool. We also use non-contact, non-surgical, laser-induced retinal degeneration model to screen drugs. Getting back to the memory, which is the focus of this talk, and as just to remind you again, we're going to focus on what we know, what we have contributed, and how we can bring in the non-drug approach to the field, because this field is known for failed clinical trials. We still do not have another drug, anti-Alzheimer drug or any of the anti-memory loss drug. So for those who are from outside neurology, we need in order to learn something, we need to be able to pay attention to it and then rehearse it or review it. So it is encapsulated in this scheme of having an encoding capacity and being able to consolidate what is known as long-term potentiation, a very important pathway of active synaptic plasticity that offers brain the experiences in the form of molecular messengers that become strong learning and memory packets in the brain, and then being able to retrieve it. If you're not able to retrieve it, it's a philosophical question. If we are not able to retrieve it, does the memory still stay there and can we retrieve it at some other stage? But retrieval is an important aspect as far as neurobehavioral studies are also concerned. If you remember, I mentioned neurobehavioral assays, such as the Morris-Water maze. So it not only analyzes new memories that are formed, but also the capacity to retrieve by what is called as a retrieval test. Some of us may know and some may not that short-term memories are allocated to certain parts of the brain, neocortex, prefrontal cortex, and the long-term memories, if they are rehearsed and reviewed by some of the chemical messengers that are known to underlie learning and memory, can be retrieved from hippocampus. So research in our lab has focused on hippocampus. We do a lot of hippocampal research, spatial memory research with relation to water maze. Amygdala, of course, adds the emotional weight to the memories, and that helps you to associate memories and to recall it more promptly. Some glimpse of how what is happening inside presynaptic and postsynaptic terminals. Action potential is fired and the vesicles loaded with chemical neurotransmitters are driven to the synaptic membrane, to the synaptic cleft is where they release the neurotransmitters, acetylcholine, serotonin. They either diffuse out or they are reuptake. They can be reuptaken. And this is exactly where the drugs act. The drugs act at inhibiting the reuptake, and they are called, as you know, acetylcholinesterase inhibitors. And this is where the farthest we have gone with regard to management of patients of Alzheimer or Parkinson's disease, related dementia or vascular dementia or ALS, FTD, as you may call it. In fact, even drug-induced amnesia. So these are molecules that are involved and have been were first proposed, as you may know, by Eric Kandel, who won a Nobel back in 2001, I think, for his work on aplasia. And if you remember the siphon and the Gale experiment, the sensitization experiment, and these molecules such as CRAB, which is the transcription factor for activating new gene transcription, or MAP kinase, PKC, PKA, and these are some of the important molecules, molecular messengers that are involved in learning and memory. None of these molecules have been studied in yoga intervention studies, for example, in people who are having a pre-Alzheimer disease state, such as mild cognitive impairment. This is, I think, a very good candidate to study and not only go as far as just looking at semi-quantitative measures such as neuropsychology assessment or memory tests or quality of life tests or SLCT or so many other psychology related tests that we routinely go about doing when we design our yoga studies. Much of what probably needs to be done is what the drug companies are doing, actually looking at these molecules, the MAP kinase, the PKA, the PKC, the A beta levels, because these are the markers or the drivers of learning and memory as they are formed as packets in the different anatomical locations that I just described. We can easily describe, memory loss into reversible and irreversible. Aging is, of course, irreversible. Disorders such as Alzheimer and vascular dementia, FTD, Lewy body dementia such as PDD are irreversible. Most of them, as we stand today with our current knowledge of medicine, medical advancement. Others such as trauma or those related to hypothyroidism or infection probably can be reversed. And again, from the kaleidoscope of yoga meditation research, we need to identify where we can easily work on things probably which are irreversible, and at the level where they're not reversible at the management level. So till now, we pretty much know from what I have talked about and what you already know from your residual knowledge in neuroscience, from a graduate studies and otherwise, that synaptic plasticity is the hallmark of learning and memory. And the molecular signaling cascades, as I showed in those slides, are very important for long-term potentiation and long-term memory formation. And this happens at the mRNA level, as you said, as if you remember, I showed the postsynaptic CRAB level, the transcription factor, which gets activated when the presynaptic terminals activate. The vesicles will go on to release the neurotransmitters in the cleft, which are taken up by the NMDA and the AMPA receptors in the postsynaptic neuron. And then the cascade of molecular machinery gets activated, and which leads to new mRNAs and new proteins. The new proteins that are formed are actually the bundles of memory that Kendall proposed are the basic mechanisms of memory formation. This is him and his team that won the Nobel in 2000. I stand corrected, not 2001. These were his experiments of Gill response from siphon sensitization, and he basically showed in this busy slide, you don't need to worry too much about, basically talking about the importance of PKC being recruited by CAMP and then formation of more MAP kinases and PK in the nucleus. As a result, the CREB inhibition was rescued, and the CREB activation led to more PKC formation, which started a cycle of new protein synthesis, which leads to memory consolidation, retrieval, and so on. Memories that are just if you can classify them, can be genetic. ApoE is one such gene, which is a lipid transporter, and some people are more vulnerable. There are different subunits, epsilon 1, 2, 3, and some people are more predisposed to that. I do not know how much our non-drug approach, such as yoga meditation, can alleviate this problem, but certainly at the cellular level, where the impact comes from amyloid beta deposit, and talk about phosphorylation of certain proteins, how they can be handled, some new bodies, if they can be handled. Immunological aspects, if we can address those aspects of memory loss, if we can address them with our yoga intervention studies. And of course, the vascular, the related memory loss, such as stroke-induced amnesia, are something which a considerable amount of them can be addressed by risk reduction, such as you know, by lifestyle modification. Those are possible by simple exercises, daily yoga practice or meditation. Some of these studies provide a scaffold of how most of us study these research publications and present them in our journal clubs. This is how the scheme of things look like. We will take a cell culture, we will stimulate them with LPS, and then study the rescue of the LPS-induced inflammation. Markers that are famous and are more fashionable to study are often TNF-alpha, IL-6. So, and this was another study that showed that once you induce such cells with LPS, which induces an inflammatory cascade, you can show that this kind of study reflects at the neurobehavioral level also, that this study, this particular study showed, for example, that the escape latency time analyzed by water maze was reduced if the LPS was not given versus when LPS was given. So LPS inflicted an injury, it inflicted memory loss, and the amount of time the mouse in the swimming pool water maze, called the water maze, took time to use a submerged platform using the spatial cues in the room was less if the LPS was not given. It was more if LPS was given. And if you add your agent of discovery, it could be a herb, it could be yoga practitioners' blood-derived stem cells, it could be any of what you want to test. You could use the same assay and the same scheme of methodology to answer a simple question of how much a yoga practitioner's blood can rescue the memory loss induced by LPS. In this case, that particular group was lacking. It was only LPS versus control. And they showed that when you put an LPS, the memory loss happened. When you put in LPS, the amount of beta deposits increased. So all the parameters of memory loss or parameters of Alzheimer's disease seem to mimic in this particular cellular model. And this is what the entire story is about. To go back to Alzheimer's disease, we know amyloid beta is a culprit. I should have brought the slide earlier, but I assume a lot of us know that Alzheimer's disease is characterized by amyloid beta deposits. They can be cytotoxic. They can lead to amyloid plaques, and that can interfere in synaptoplasticity. And this can also be further aggravated by microglia and the astrocyte activation as well. This is shown here in this slide. And there is the tau pathogenesis that has also come up because it's more intracellular. Amyloid beta is extracellular. So for the intracellular amyloid beta, there are hardly any studies where the MCI or the mild cognitive impairment or the pre-Alzheimer's disease state or pre-memory loss state, if screened from community, could be given a lifestyle intervention study and their amyloid beta could have been studied, or a cellular system where a certain cell line which has the features of neuronal cell line would have been studied by inducing LPS versus drugs such as cholinesterase inhibitor and a yoga or a meditator's blood, which could also be used to see what are the profiles as compared to LPS, as compared to cholinesterase inhibitor. These kind of comparative effectiveness studies are really lacking not only at the clinical level, but even at the preclinical level. Bala was telling me there is a center at Harvard, a patient-centric center which looks at head-to-head studies on various drugs of which is best for patients for a certain disease. This is what I gathered from you. But I think this is lacking mostly in other global centers around the world, and so much prominently lacking in our yoga and meditation related studies. Same, the philosophy applies, and the same argument applies to the tau pathogenesis. This happens due to hyperphosphorylation of tau, which may do similar havoc on postsynaptic neurons, causing mitochondrial dysfunction. Synaptogenesis is altered and then compromised, if analyzed at the animal level, and also at the level of electrical patch clamp studies that are done in the molecular biology. Similar kind of dementia that I mentioned in the beginning also visits clinical states such as ALS, FTD, also Parkinson's disease. They vary. The only difference is the culprit is not just amyloid beta or neurofibrillary tangles. There is RNA metabolism disruption, that machinery is affected, or TDP43, one of the protein which is the culprit in ALS, similarly alters the synaptogenesis and causes ALS frontotemporal dementia. And the culprit is similar for ALS as well as the FTD, which is TDP43. And again, neither the TDP-43 has been analyzed in ALS or for Parkinson's disease related dementia in terms of recruiting them as candidates for our any of the lifestyle intervention studies, exercise intervention studies, yoga or meditation intervention studies. The only thing is we need to look at the TDP-43 levels. We need to get the CSF through proper ethical committee approvals and informed consent if possible. Or even a lot of what data we have shown at the blood and serum level can also be recapitulated and organized to answer the simple question of yoga and meditation intervention studies as to what is the outcome in one of these downstream and upstream mechanisms of TDP-43. So that may have some implications for not just Alzheimer's disease or pre-Alzheimer's disease, as I said, MCI, but also Parkinson's and ALS. So similar thing applies to stroke. We know that the vascular supply is essential, and if it is not there, and if there is reperfusion, it may cause ROS activation. Free radical damage could exacerbate and aggravate the entire problem, which can implicate memory loss. And the stroke rehabilitation program, I think there is a lot of work already happening with relation to at least from the breathing techniques and meditation. But I will skip this to bring this slide. I thought my colleague Bala is from the anesthesia and used lots of drugs and chemicals. There are those drug-induced cognitive loss. We were walking around the campus. We mentioned that a lot of people, up to 40%, have cognitive loss when they undergo anesthesia, and about 2 to 3% may have long-term cognitive decline. So they follow, they have their pathways all lined up, and that is very well known either at the level of NF-kappa B or at the level of ROS activation, or the level of the molecules that Dr. Kendall had proposed, and I molecules, calmodulin, CAM kinase, PKA, PKC, all of them also are affected in case of drug-induced MS. And I will show you in subsequent slides, we used some of these agents, these amnesic agents, to cause amnesia in the mouse, and how we rescued it using an Indian herb is what is going to be focus of the next subsequent slide. But before that, let me highlight the importance, and this is already highlighted in the entertainment industry. This is an Indian movie, Black, that highlights the burden and the tragedy of Alzheimer's disease. This is the movie Black, if you have time. And this is another movie, Sweetheart, another Hindi movie, if you might get your hands on this movie. The translations are available. What is the plight of average middle class individual living in a developing world, and might apply to some of us in the US. Or how much is the burden, economic, socio-economic burden, or the health burden, health cost of health burden on diseases that are preventable. In Sweetheart, this particular movie, this couple were famous for doing some mean jobs to get money to be able to pay for angioplasty. And I think the same thing applies for cardiology related cognitive deficits or Alzheimer's disease as a whole. So I just wanted to highlight this burden is immense in the US also. And most of that, to my knowledge, is also to a large proportion preventable by very simple lifestyle changes. And to how to make it happen, how to systematize and institutionalize this in a medical healthcare system is a question me and Mala were discussing, and all of us should probably deliberate on this. So we all agree, and as I also mentioned in the beginning, there is no treatment for Alzheimer's, and whatever is available is cholinesterase inhibitors, which work at the reuptake level, inhibiting the reuptake and basically releasing more and more acetylcholine at the synaptic cleft level. The other things that are coming up are either lifestyle intervention, exercise intervention. Those studies are coming up. Some people are going with stem cells, and it has limited potential. Some herbal remedies, some Indian and Chinese drugs are being attempted. I'll show our data with one of the Indian drugs. There are some reports that try to predict Alzheimer's disease early by looking at retinal amyloid deposits, which is a little earlier than before the Alzheimer's really sets in. And people are trying music and other light and sound. Somebody at the MIT is working on working on light and sound intervention in patients' Alzheimer's disease. All kinds of non-drug approaches are now coming up. And this actually opens up a new vista for us to take this up as a thrust area, maybe for the Center for Consciousness already set up in this particular university, Harvard University. We about 10 years back started thinking of the same problem of looming failed clinical trials in Alzheimer's disease and other memory loss clinical situations, some of diseases that I mentioned. And it's thought that we should try to use the same methodology, the same systems that are used in a wet lab to study what was already known in Indian traditional medicine called the Ayurveda. Brahmi is one such drug. It's called the Bacopa monnieri, which is a botanical name. It is rich in bacosides, and it is already well published. It is published in the Indian ancient texts. So we took these extracts and we used three kinds of drugs: the NMDA receptor antagonist, the NOS inhibitor, and the anticholinergic drug to cause amnesia. And we used it using the same scheme of Morris Watermaze. And we use this scheme. It's a 7-day protocol. We gave this orally, we gave the amnesic drug intraperitoneal to these mice and made sure that they caused amnesia, and we could assess them in the Morris Watermaze. So there was a loss of memory, which we could see in this. I did not bring a picture of a circular tank, which is actually full of water and then artificially divided into 4 quadrants. And there are spatial cues in the room, as I was explaining. And as the mouse tries to, and there is a submerged platform which is below the meniscus of the water, that the mouse tries to not get drowned and look for it as a rescue, not to drown. And the amount of time it takes from the way it is, the way the mouse is put into the water to the submerged platform using the spatial cues is called the escape latency time. So the sooner it reaches, the more the good the memory, and the later it reaches, the less the memory. Use an amnesic agent such as the one we used in our scheme of things, namely MK81 for NMDA receptor, known to be and proposed by Rick Kendall as one of the receptor for learning and memory. And we used LNNA for NOS inhibitor, another molecule used in learning and memory. And the third, the anticholinergic drug, scopolamine, to again get rid of the memory learning capacity of the mice. And we found each time we gave this drug, the memory loss was more pronounced. So it was pronounced more than the control, which was just a vehicle given for this kind of these 3 drugs. So we were able to show that once these mice were amnesic, they lost their memories, were not able to learn. Every time we gave increasing dose of Brahmi or Bacopa monnieri, it was able to rescue, as seen in this graph, the amnesic agent caused memory loss. So we were able to show that the Brahmi or Bacopa monnieri was able to rescue the lost memories artificially induced by these 3 amnesic drugs using the Morris Watermaze scheme. Now we are doing the similar kind of study using about 100 yoga practitioners given common yoga protocol for 3 months, and then collecting their blood, and then the lineage negative peripheral blood derived lineage negative stem cells, then putting them in the same model, comparing them with Bacopa monnieri, Brahmi, and also using the same scheme of amnesic agents. So this is what where we need to take a yoga and meditation research. If we really want to have substantive and very strong, robust molecular evidence to be able to prescribe yoga and meditation protocols for various diseases, in this particular case, memory loss. This is another of the brain homogeneities we took out from the mouse that were subjected to amnesic agents and Brahmi in those 2 control groups. And of course, there were other control groups such as sham and vehicle, etc. And we found that calmodulin actually is also significantly increased, as was shown by Kendall in his own experiment. So to the level donazepam or rivastigmine that is given the clinicians to their patients, to the same extent, at least at the preclinical level, we were able to show. And these are just studies. I'll skip them. This particular study shows the retrieval part of the memory, which I tried to talk about right in the beginning. So one is the acquisition, the other is the retrieval. But from this particular drug, Indian herbal drug, was able to rescue the memories, lost memories caused due to amnesic agents in a similar fashion as the conventional drugs used in the clinic. This pertains to this long-term potentiation phenomena, the strengthening that I mentioned right in the beginning. There are now clinical trials from Brahmi, which pretty much substantiate what I'm talking about. This is actually happening in the field of at least the Ayurveda or Indian traditional drug. So the things have gone from preclinical molecular studies to LTP level to now at the clinical level. So this is where we can now propose to have Brachypneumonia or Brahmi, at least we're trying to get this prescribed by the Indian doctors, Indian neurologists in patients of Alzheimer's disease and also in stroke patients, ischemia induced. We did another study where we wanted to look at stem cells and how they fare with respect to such a animal model of memory loss. Lineage negative stem cells are those that have got their fate or lined up, and they can be easily screened from max column magnetic stem cell. There is a magnetic stem cell sorting facility that is available. You can get rid of the markers that assign lineage to a stem cell, and you make them lineage negative. We had some previous work in a similar retinal degeneration mouse model where we had shown that these lineage negative stem cells actually go to the laser spots into the mouse eye, and they're able to home on and get mobilized to that state of injury. So we thought we should use it for our memory mouse model also. The same scheme of modus operandi is of first injuring the mouse with intrahippocampal stereotactically injected into the mouse. Of all the memory. Tested this and got amazed, and then subsequently gave it the versus vehicle versus sham injury, those kind of controls, and then looked at how much this could rescue the lost memories caused due to amyloid beta. Why amyloid beta? Because I already mentioned in the first few slides, it's the hallmark of Alzheimer's disease and pretty much for Alzheimer's. And also a good model for brain injury as a whole, if not just for Alzheimer's disease, because it's, after all, you are assaulting the intrahippocampal, you are assaulting the hippocampus, which is the store of long-term memories. So in this scheme of things, we got the stem cell isolated, screened for characterize using this paraphernalia that we use in standard neurobehavioral or a molecular biology lab, namely water maze, rotarod, organization, FACS, and isolated from the umbilical cord, and then using it for, after sorting it out, using it to cause injuries. And of course, characterizing these cells that they are of the kind that we actually wanted to transplant, putting the injury into the brain. These are some studies to validate that the injury has happened in the mouse hippocampally, and then to see real results at the behavioral level. If the memory that was lost due to amyloid beta deposit was rescued, as you can see here in the first particular slide. If you see A, kind of becomes, because of contrast, you can't see it. But if you look at A, those are the escape latency time over 1 to 6 days. As the mice learns, obviously it reaches there sooner. There's a merged platform because learning. And as you can see, the amnesic agent is there, the amyloid beta injury group. If you see, there is no learning, and the curve is flat. But when we put the lineage negative stem cells, the learning was rescued or resumed. So we were able to show that these cells are able to resume the lost memories caused due to similar Alzheimer's disease kind of a situation. But the important thing here is that we have the source. What is the source? The source is umbilical cord blood. Right? We isolated. This is the procedure. So the study that we are now doing, you might like to take up, is to use what we do basically is to engage and recruit after using informed consent the pregnant women for third and fourth week of 4 week of yoga intervention. The third trimester has a certain particular protocol, and the fourth trimester is obviously more breathing oriented, not so many asanas and physical postures. And we collect the cord blood just like this particular case. Now we are comparing our studies of what lineage negative stem cells, to the extent that they were able to revive the lost memories, how they compare with pregnant women who actually underwent yoga and meditation. And we collected a similar umbilical cord blood from them and isolated lineage negative stem cells from those yoga practicing pregnant women, and not only looked at the yield of stem cells, but also are now testing as part of our PhD students' work of how much, to what extent the lost memory in the animal model can be rescued as compared to those who are non-practicing. So these are the experiments where we need to go into to take the non-drug approach, a non-pharmacological approach to a level where the integration with the modern medicine is seamless and unquestionable. And of course, if it really shows the results. So these were just some of the characterization of the cells, and these are more complicated studies about a web camera using the the pathway a mouse uses, just to kind of normalize to the motor ability of the mice, which may vary from one mice to the other. Some may be more agile and might reach the platform early, giving us a wrong impression that the mouse is actually learning faster, but actually maybe using a longer route. So these are just those of you not familiar, these are additional cues, additional when you do an ultrasound, you do CT and an MRI. So it's an additional behavioral parameter to make sure to basically substantiate the result that I'm calling off here, showing you here. And the traditional strategy we know in all molecular experiments is to use an inhibitor. So if you believe that BDNF is actually involved, as we proposed, and CREB is actually involved in lineage negative stem cells rescuing the lost memories, we need to get rid of that by using inhibitors for CREB, inhibitors for BDNF, which is what we use. And these again, if you focus on A and D again, A is the behavioral and D is the molecular aspect of the same set of experiments. But it tells us as soon as the inhibitor for BDNF is used, the resumed memories are lost again. And so is the case if you look at the PRK inhibitor, which is another BDNF inhibitor, the memories or the amount of escape latency time that was there. It's a quantitative call out from this is another measure, as I mentioned. I'll skip this. I'll skip all of this. Is basically looking for other growth factors and cytokines that may be involved and maybe basically mediating the rescue of lost memories, such as CNTF, GDNF, BDNF, and so all of them. If you go into the details of that by knocking off each of those genes, either by creating knockouts for those in the form of knockout mouse, or by siRNA technology, or by using inhibitors, one can easily assign the gene a function or the molecular function, and also where it falls in the pathway that is involved in learning and memory. And in this case, lineage negative induced memory revival. So, the last part of my talk is now focused on what I've been telling in my entire talk of trying to give you a flavor of what do we know about learning and memory, what we have done, and the clinical spectrum of diseases where we encounter dementia. And then to tell you what I was telling, and now to in the next 3-4 slides to again highlight the importance of bringing the lifestyle intervention and yoga meditation studies to the same level, the same methodological rigor that I just presented, those studies that are all in the public domain and PubMed. Which is also the reason I highlight is because this is not happening with related to yoga. This is happening in certain levels, in certain studies. And for example, a lot is happening in high altitude related research, a lot is happening in meditation research, but a lot more needs to be done at the molecular level, which is probably lacking. Now, to those who are not familiar with yoga, I believe there are a few. We know that yoga is not just physical postures, it is not just meditation. In fact, yoga and meditation are also not compartmentalized or dissociated from each other. Yoga actually is a 8-limb path. It encompasses the social and the moral doctrines called the yama and the niyamas, followed by the physical posture, which are the pranayama, the asanas. That readies the body for a good breathing technique or pranayama. They are known by different names. You can call it by Rom Vilom, you can call it by Ujjayi, it can be called by Isha Kriya. There's a variety of Sudarshan Kriya and many types of pranayama. And the 5th step, the process of enriching internally, or sort of withdrawing your senses from the outer world to focusing for inner enrichment, is called the Pratyahara. Then the Dharana, Dhyan, Samadhi are the last 3 steps, which is essentially the meditation. So yoga basically is very much a part of physical posture, social, moral indoctrination, or self-regulation, if you could call it, and then physical and breathing techniques and the meditation. So this in together, it is a package. It's a module which needs to be protocolized in whatever fashion, oriented towards asthma, towards memory loss, towards diabetes, or whatever that one wants to fashion a study and design a clinical study. And I will come to this later. So we have, for example, some of our studies have gone that far. So this particular, first on your left is a study which we did for common yoga protocol that we all practice on 21st of June, the International Day of Yoga. It's a 45-minute yoga protocol encompassing lying down, prone, standing and sitting asanas, and also the breathing techniques and meditation. And what we did was a 1-month study, and we looked at not only many other biochemical and molecular parameters, but here to show you some of tail A and tail B tests that measure the cognitive processing, so which was significantly increased in subject that went through a common yoga protocol. This is not a randomized controlled trial, so it's a pre and post study for 1 month. Just came out in Frontiers in Public Health. Another study that we did, we flew some people up to the high altitude depleted of oxygen content, and related sickness is something very well known, and there's hardly anything in terms of medical intervention other than bringing the patient or the person down to a low height or low altitude, which we have now here. So for that, we took these people up and just looked at PO2 level, looked at their cognitive processing, their puzzle solving ability, their memory, their quality of life, and other parameters. And we found that most importantly, that the PO2 levels went up in this group of people who actually underwent this short breathing technique for 4 days versus those that did not undergo this. And not only the PO2 levels went up sooner than the others, and to more proportion this particular breathing technique. But I opened the doors and I shut them very quickly to the door of something called Prakriti, or what is described in Indian traditional medicine as Ayurvedic principles of clinical, how would I put it, clinical phenotype. Kapha, Vata, Pitta as they are called, and there are subtypes of Kapha, Vata, Pitta. And we showed the particular Pitta kind of, a particular Indian Ayurvedic clinical phenotype, Pitta Prakriti individuals were able to rescue PO2 in a more superior fashion than those who were of the other Prakriti. So, I feel as a scientist and as a para-health worker that this is so important for us, our duty to not keep the population and patients away from this knowledge, which can benefit them in prevention and rescuing them from a certain disease that they may encounter in their lives. With this common yoga protocol, another part of a PhD student study, Kanupriya, actually on your left, I showed you briefly in the earlier one, pre and post study of the common yoga protocol, increasing cognitive processing. In her thesis, this was a randomized control trial of the same protocol, common yoga protocol, and we showed that it actually increases BDNF and VEGF levels also. Yeah, so just ELISA results. And VDNF, as I showed, if you remember my presynaptic, postsynaptic terminals and synaptic plasticity and my narration of facts about the Candle's experiments, PK, PKC, calmodulin, and BDNF was sitting as a receptor shoulder to shoulder with AMPA receptor in the postsynaptic terminal. So BDNF is what is increased if you just practice common yoga protocol. Isn't that amazing? So this kind of work is happening all around us, but we need to further ramp it up, maybe as a global community of people who fear for and who care for people who do not have enough resources to pay for their different diseases they may encounter. So best would be to use this as a means of intervention in addition to usual care. Maybe in certain cases, what I like to tell in every talk is to screen people at the pre-disease state. For example, as I mentioned, pre-Alzheimer is MCI, pre-wet AMD is dry AMDs, pre-diabetes is pre-diabetes, cancer is pre-cancer, like stroke is PIA. So if we can get hold of these TIAs and dry AMD and pre-diabetic and precancerous and dry AMD, MCI, this is where we should focus and then study intervention, lifestyle intervention such as yoga intervention, and can we halt the conversion into fully blown cancer, fully blown wet AMD, fully blown diabetes, fully blown Alzheimer, fully blown stroke. With that, I'll end my talk. I'll be happy to take questions. Thank you very much. Nice one. The common yoga protocol is something that was arrived at by what is called as a Delphi protocol of Government of India Ministry of Ayush's effort to put in the yoga masters, the scientists, the endocrinologists, physicians together and arrive at the most preferred and standard yoga protocol that would be fit for all ages, gender, and from kids to elderly, and which could encompass not only the ancient knowledge of yoga, which is the 8-limb path, the Yam Niyam, Asana, and Samadhi, as well as the published evidence from people who have published. So these are basically some standing asanas, some sitting asanas, lying down asanas, prone asanas, 45 minutes. Yeah, every 45 minutes. Under supervision, yes. So this is all non-Zoom or non-online, in-person studies. And this is, we all practice on the 21st of June all over the world. Now, specifically designed for International Day of Yoga. But because it was standardized and it went through a Delphi protocol and apparently had a lot of 10, 12 rounds of deliberations. So because it was being practiced globally, at least on the International Day of Yoga, so we thought this would be fit to use it. We picked it up from healthy volunteers. I didn't show any data of this being used for MCI. I only showed data from healthy individuals. Even in the healthy individuals, the VEGF and the BDNF levels went up. If I recall correctly, 3 times. Didn't do that kind of comparison, but I'll review that literature. We didn't have that arm also in that particular case. Good to see you, Rich. Oh yeah, thank you. Thank you for reminding me. This should have been my last slide. This is what we are aiming for, to bring the best of whatever data that I present or whatever I try to talk about and argue for is this kind of a healthcare system where we have as General Hospital or PGI or whatever best hospitals in the world, or even district hospitals, where we have professors of anesthesia, professors of Ayurveda, professors of medicine, professors of neurology, professors of yoga, working hand in hand. And so there is a triad system where based on their clinical spectrum, whatever that they present, they could be referred to a yoga professor for particular yoga, combined with that or not combined with medical medicine, what he or she wants to treat or not treat. So this presents a system where we could use the same paraphernalia present in big hospitals, MRI, CT, semi-auto analyzers, ultrasounds, or whatever infrastructure we have, to be best used for launching not only launching services in parallel, but also from that service, whatever data we get from the interventions of yoga, interventions of Ayurveda, interventions of modern medicine, for example, for diabetes, you could have comparative effectiveness studies done seamlessly, and as we practice, that data will come and will further strengthen the system of the integrative health system, where we bring in the evidence-based best of East and West. That's the yes, it is. This was Sudarshan Kriya. 0 day. A lot of people asked if you allow people just landed at 10,000 feet. This was a study at Leh Ladakh and a place called Mansarovar, one of the pilgrimage centers now in China, Tibet. A lot of Indians go there. So it's about 14,000 feet. So we did it right from the time they landed. Oxygen saturation, LDL, ran those quality of life tests, SLCT, mental well-being tests. Some of the data some of the samples are already preserved with us. Yes, somehow the person who was involved left, so we are now going to be analyzing the various other, many other markers. Some questions?
Ask in humans, what are the biomarkers for memory that we found?
BDNF was the one that I showed. This was a question that was just now asked, but we did not take patients of Alzheimer's disease or mild cognitive impairment. We just took healthy volunteers, healthy individuals analyzed and screened by GHQ, and we found that BDNF levels were increased. Were correlated to those found when we exercise or we jog. So I have I did not compare that. So I don't know. I'll come back to you later. I believe we have to, collaborate more actively with the neurologists and use tests that are used by them for various diseases. One of them is the psychology battery, the cognition related tests, and other tests. The markers that are known to be of diagnostic value are acceptable to the world of neurologists is amyloid beta. That is one. Also, BDNF not accepted, but yes, there are studies that it has a neuroprotective mechanism. Pre-postsynaptic strengthening is empowered by BDNF levels. Also, to use more and more neuroimaging studies. There are a lot of studies already available with meditators looking gray matter. And I guess we have to work harder towards evolving more robust means of translating the molecular biology studies. For example, the one that I was telling again and again, PK, PKC, calmodulin. I'm pretty sure we can't measure this in the serum and plasma. We need to have. I think of an opportunity where we can some evidence, versus Parkinson's versus Alzheimer versus Parkinson's plus yoga, Alzheimer plus yoga. Carrying out EEG studies, look at the alpha, beta, theta waves. That is another measure. It's one indirect measure. Yeah, but this is very well known. For the reason that probably causes some confusion in your mind is because of the famous nun study where there was a lot of amyloid plaques found in their brains, but yet there were reserves of memory and they were carrying on with their functions. But to the extent that the amyloid beta can impinge on the hippocampal function is very well demonstrated even by our study. The moment you put stereotactically, you deliver it to the mouse brain, it stops learning. And you can see the ELT going up a straight line. We saw it for scopolamine, we saw it for 801 and LNN, and we also saw it for the amyloid beta also. For the purposes of defining and redefining Alzheimer's disease, yes, amyloid beta, one hypothesis is that, tau is another, some people say RODS. So that's changing, and of course, factorial, ApoE is another, so genetic aspects. Causes can be many, but the eventual symptom is memory loss. So if we were to take the nun study as a substrate for hope for tomorrow, for at least we have hope, even though those nuns had huge amyloid beta reserve in their brains, yet they were able to show normal learning in them. So something was there which was enabling normal working memory to functionalize, to be functional. I believe we can do this by yoga meditation. All we need to do to arrive at this particular conclusion is to do such studies. As we do, we will never know. I'm proposing a good solution that let's take the subject at the pre-disease level, at the MCI level, and then give the intervention and study the conversion rate versus those who did not undergo the intervention. So there was a study launched by one of the preeminent yoga institute in India. Your question is how many people, what population of India do practice yoga, is it? So that particular study was launched by this institute called S. Vyasa. So I was a collaborator and they asked about 150,000 or so. I don't know how many of you practice yoga. So the question was not really yoga, it was divided into whether you do pranayam, because the definition of yoga itself, as we all agree, is some people may be doing physical posture, yoga. Some may be just doing breathing techniques and it is yoga. Chanting itself may be yoga. Different definitions. So they asked, are you doing pranayam? Are you doing yoga? Second question. And 3, are you doing physical postures? 4th, are you meditating? So to these questions, the answers of 1,000 or so odd people that were sampled, 10 to 11% across different zones of India. I think that we will agree matches to American study. It's higher here, so it's. But I would like to do another study, a more organized, a more systematic, with the help of more study when it was already launched. So it had a certain amount of rigor, but more. Oh, it's a very, very, very good question. I'll answer this by our own experimental results with Brahmi. And in fact, the study, preclinical study of Brahmi, Bacopa monnieri. If you remember the slides that I showed when we used amnesic agents such as scopolamine, LNNA, and MK801, we were able to deplete memories and show it in neurobehavioral assay for Morris Waterman's. Only when the memories were lost, we were able to recover or rescue the memory loss with different doses of Brahmi. There was another group called Brahmi per se group, amnesia caused, but in those mouse we were not able to show, at least to the extent, to the mouse level, we were not able to show a further increase in memory. Displayed by Morris Waterman's. To the questions about Brahmi in clinical use, I showed some trials. I'm not a neurologist, so I do not see patients, I do not know. But from anecdotal interactions and questions, my interactions with my, for example, neurology head, my former head, he used to tell that he could see many people, many students who were taking what is in India called a kind of a them, and he probably gave to 6-7 of his family members this Bacopa model, this Brahmi, and he found they could retain better, they could sense more, and they were less anxious. Have that answer. I gave it to my daughter who went for IIT, and my daughter also found it was. I mean, this is not scientific. This is what she told me. I mean, this is what our data shows. Yes, Queen. Would say I would leave it to Bala. Bala, why don't you organize a Novat? Why don't you organize a yoga camp for the Novartis top management? PGI Chandigarh, we have this yoga center, and what we are doing is we are not so much bothered about the patients at this moment, the first year. All we are focusing is on the doctors. Just come and do yoga. And there have been studies, I don't know if you're aware of people actually it was a study around the residents, medical residents, who were given yoga versus another group, yoga plus theory of yoga. The experience of yoga and just the knowledge of yoga. Is comparative effectiveness the answer? If you do a metformin versus diabetic yoga protocol and see the results? These kind of studies.
Yeah, that's comparative.
Need an FDA approval. Not a drug. Thank you.
So thank you again, Dr. Anand, for joining us today and sharing some of your wisdom and work on this amazing topic. So for those of you who are interested in joining us for future speaker series, our next upcoming speaker series in November is happening on Wednesday, November 16th. This will be solely online. We'll be joined by Jonathan Dinsmore, who is a master's candidate in psychological science at the University of Tennessee in Chattanooga, and he'll be talking about mysticism and modern science, and specifically a scale on mysticism that he's developed with his mentor. So we're excited to learn more about this topic, which has information on how you can stay in touch with us. You can join our mailing list and sign up for updates with the QR code on the bottom left corner. And we hope to hear from you soon, and we hope we continue to engage with you as part of our community. Thank you so much.